Hello, good morning. Myself, I am BSR Botany lecturer. Today, our topic anaerobic respiration. Already, aerobic respiration completed. Anaerobic respiration. Anaerobic respiration is nothing but fermentation. The term fermentation was coined by Louis Pasteur. This process explained by Gay-Lussac. In aerobic respiration, the glucose completely oxidized and synthesizing the ATP. In anaerobic respiration, that glucose converted into pyruvic acid, then the pyruvic acid fermented to ethyl alcohol. That is the process. The end product of the glycolysis, two molecules of pyruvic acid. Now the two molecules of pyruvic acid undergo decarboxylation. So when it undergo decarboxylation, two molecules of acetaldehyde forms. In each acetaldehyde, two carbons are present. So for that, the enzyme is pyruvic decarboxylase or pyruvate decarboxylase. So now these two molecules of acetaldehyde undergo reduction. So reduction, hydrogen donor is NADH2. So two NADH2s are converted into two NADs, acetaldehyde fermented to ethyl alcohol. So here very important, when glucose converted into ethyl alcohol, the result you observe. In glycolysis, how many NADH2s are synthesizing? Two NADH2s. How many ATPs are synthesizing? Four. In that four or two are consumed. So net is 2 ATP. So here acetaldehyde reduced to ethyl alcohol, for that two NADH2s are required. So these two NADH2s are formed in the glycolysis. So it means these two NADH2s are also utilized. It means anaerobic respiration, finally, what is the result? Only 2 ATP can be synthesized. Not only that process, just you observe another lactic acid formation. Same thing, the pyruvic acid undergo reduction to form lactic acid in presence of lactic dehydrogenase. For that, two NADH2s are required. The two NADH2s are formed in the glycolysis only. So in anaerobic respiration, the final result of ATP only 2 ATP. So this is the anaerobic respiration. Then when you go for respiratory quotient explains during the respiration the ratio between number of moles of carbon dioxide evolved and number of moles of oxygen observed. That is called respiratory quotient. So the respiratory quotient means the ratio between carbon dioxide evolved and oxygen observed during respiration is called respiratory quotient. Here important thing, you observe in the carbohydrates, respiratory quotient is 1. In fats, respiratory quotient is less than 1. Whereas in organic acids, respiratory quotient is more than 1. Because look at their carbohydrate. Example, glucose. C6H12O6. Carbon number and oxygen number same. So that is why for the oxidation of this glucose, same number of oxygen molecules are required. So carbon number and oxygen number for oxidation same. So that is why respiratory quotient is 1. Whereas when you go to fats, you just observe here carbon number is 51, oxygen number is 6. So that is why oxidation number for oxidation, oxygens are more are required. So that is why respiratory quotient is less than 1. When more oxygens are required for oxidation, there always respiratory quotient is less than 1. So that is why in fats less than 1. Example, tripalmicin. Whereas when you go to organic acid, anything that is malic acid you take, there carbon number is 4, oxygen number is more. So C4O5. So that is why for oxidation of this substance, less oxygens are required. So respiratory quotient is more. So that is why 4 by 3. So respiratory quotient is more than 1 in organic acids. So this is the respiratory quotient, and the respiration process is over. Thank you to all.